Hi, my name's Ross Dickens. Thanks for coming along this afternoon for Matt Witkowski's PhD completion seminar. So, Matt and I first met back in 2010 at the WEHI football match when Matt was a Europe student in Doug Hilton's lab. And apparently I made quite an impression on him on the football field, so he came and asked whether he could do an honours year with me, which he did in 2011. Matt topped the honours year that year, and I was really thrilled when he decided to stay on in the lab to start his PhD, which he did from 2012 through till this year, and he's writing his PhD thesis as we speak, and we'll submit probably in a few weeks. So that's really good going. He's knocked it over in 3 years, and that really, I think, illustrates one of Matt's qualities, which is a real high-intensity individual in everything that he does, not only at a personal relationship perspective, but from a scientific perspective. I've had a great pleasure in interacting with the guy for the last 4 or 5 years, watching him grow from I guess, quite a junior scientist who was always very gifted and very technically proficient with his hands into someone who now is really a young, budding, leading scientist, I think, who's able to assimilate ideas and come up with his own ideas. And I think he's going to make a great impact in New York, which is where he's going at the end of this year to start his postdoc with Yanis Ifanis, who also works on acute leukemias. Just a couple of very short anecdotes about Matt before we hit the science. I mentioned the football. He's a fantastic sportsman, as many of you know. I think it runs in the family, maybe something to do with his half Burmese, half Polish heritage. I think there's a bit of hybrid vigour going on there. And he's also got a lot of other characteristics that are. He's obviously very friendly, interactive, charismatic. He's done science podcasts. He's done a lot of work for public relations for WEHI and also for the Leukemia Foundation, who funded his PhD scholarship for the last 3 years. And as you know, he's a really good ambassador for science. So, and I'm sure he'll continue that. So, Matt, you've been a really fun guy to have around, and it's been a great pleasure working with you. And I look forward to your talk. Over to you.
There we go. Is this working now? Can you hear me? The old One Direction star. All right, great. So thanks very much for that, Ross, and thanks for everyone for coming today, particularly my mum and dad and my mates here at the front. It means a lot to have you here, so thank you. As my title suggests, today I'll be talking about... Oh, very good. That's better. Okay. Thanks, Cole. So today I'll be talking about my PhD project, which is trying to understand the role of the hematopoietic transcription factor Ikaros in acute lymphoblastic leukemia. So just in the way of background, I'll discuss acute lymphoblastic leukemia as a disease and also the role that Ikaros mutations play in this disease. I'll then discuss what Ikaros does in normal lymphopoiesis, and then go into some of the results from my PhD project. So we use a novel technique of TET-regulated RNAi to inducibly knock down Ikaros in vivo, and we've been able to explore 2 models of ALL. First one is the T-cell progenitor acute lymphoblastic leukemia, and then more recently a high-risk BCR-ABL positive subset of B-ALL. And then finally I'll finish with some future directions. So in the way of background, ALL itself is the expansion of immature lymphoblasts within the bone marrow of the patient. And this really acts to impede normal hematopoiesis, and patients often present with anemia, and due to defective white blood cell production, they're no longer able to respond to common pathogens. Now this blood smear here and bone marrow section just demonstrates the large blast-like phenotype of the ALL cells and shows that these really clog up the system of the patient. ALL itself is the most common cancer in children, and really one of the more recent findings is that the outcome of ALL seems to decrease with age. So older patients seem to respond far more poorly than the younger patients. And ALL itself can be broken up into a series of different subtypes based on either cytogenetics or expression profiles. And that's demonstrated here. This is the spectrum of childhood ALL subtypes. And you can see in red we have the T-cell progenitor ALL group, which actually increases in proportion with age. And then in blue we have the B-cell leukemia group. Now, we can see here that there are a series of cytogenetic rearrangements that will subtype this disease, and these are often prognostic. So, for example, the highlighted MLL rearrangements or patients harbouring the Philadelphia chromosome BCR-ABL have a particularly poor prognosis. And then more recently, transcriptional profiling demonstrated a group of patients with a BCR-ABL-like signature. That is, they don't actually harbour the Philadelphia chromosome, but transcriptionally they're very similar to this group. They also have an extremely poor prognosis. Now, our lab is particularly interested in transcription factor role in acute leukemias. And really, some of the seminal work genomically characterising leukemia really emanated from studies of B-cell acute lymphoblastic leukemias. So Jim Downing and Charles Mulligan in 2007 were able to do genome-wide copy number variation analysis on large cohorts of B-cell leukemia patients. And one of the major findings that they had was that genes involved in normal B-cell development are frequently mutated in BALL. So these include, and in this really basic schematic of B-cell development shown here, we see that genes like Ikaros, PAX5, and even parts of the pre-BCR are commonly deleted in this disease. And it was hypothesized that perhaps by deleting genes involved in B-cell development, you're blocking B-cell development of stages particularly susceptible to leukemic transformation. So at the time, that was the idea, and this really set off our lab in trying to model what these lesions actually do in BALL. Now, for the sake of my project, Ikaros deletions themselves only occur in around 15% of all B-cell leukemias. But what was really interesting was that in 2008, Charles Mulligan was able to demonstrate that in around 85% of this high-risk BCR-ABL positive subset of BALL, there were Ikaros deletions, suggesting that deletions in this gene were extremely important in high-risk subsets of BALL. Now, the nature of these mutations is quite interesting. Here we have a schematic of the Ikaros gene structure, and commonly there are deletions in the DNA binding domain, which generates a dominant negative isoform of Ikaros, which lacks DNA binding capacity and can also sequester away other interacting partners of Ikaros. There are also reports of mono and biallelic deletions of Ikaros, but we tend to think of mutations in Ikaros in human ALL as more a reduction in the function of the gene, so hypomorphic mutations. Finally, and quite convincingly, it's been shown that Ikaros deletions seem to predict treatment failure and relapse of acute leukemias independent of any cytogenetics. So this Kaplan-Meier curve here shows Ikaros patients where they've removed high-risk cytogenetic subsets, yet you can still see that the Ikaros-deleted patients have much higher incidence of relapse than Ikaros wild-type patients. So overall, it's clear from this that Ikaros plays a role not only in the pathogenesis of high-risk BALL, but also in therapeutic resistance. So that's from the human data. It seems pretty clear that Ikaros has a massive role in high-risk ALL, but what does the mouse tell us about the role of Ikaros in normal lymphopoiesis? And it's quite clear from the various mouse models that have been generated dating back to 1994 and onwards that these mutant models have defective B-cell, T-cell, and NK development. So they have pan defects in lymphopoiesis. And this was shown to be due to a defect in the lymphoid myeloid prime progenitor compartment of mice. And what was quite interesting is that in many of these Ikaros mutant mouse models, they actually succumb to an aggressive T-cell leukemia, which I'll get to in a moment. But that's not to say that Ikaros mutations don't have a role in normal B-cell development of the mice. If you conditionally delete Ikaros in the B lineage of mice, there's a block in B-cell differentiation. And furthermore, if you cross these hypermorphic Ikaros mutations onto a BALL-prone genetic background, you can see acceleration of the disease. So overall, this suggests that Ikaros target genes critical for tumour suppression and therapy resistance still need to be elucidated in order to explain all of these dramatic phenotypes in the mice. So this gets us to what we do in our lab. So we use a really novel technique to inducibly knock down the expression of our target gene in vivo in established leukemia often. So this is TET-regulated shRNA system, and Ross developed this in his postdoc in Scott Lowe's lab at Cold Spring Harbor. And just for the sake of this talk, I'll be talking predominantly about the TED-off system, but there's also a TED-on system I'll briefly touch on. So just to explain, you have a tissue-specific promoter here, and in the TED-off system, you actually produce an active transactivator protein, TTA. Now, in the absence of DOX in the TED-off system, you get binding to the TET-responsive element, and that results in co-expression of GFP and also your shRNA of interest. So in the TED-off system, if you leave the mice off DOX, you get target gene knockdown. And one of the major advantages of this system is that you can actually treat the mice with doxycycline, and that will inhibit the function of TTA, and therefore you get shutting off of GFP and restoration of your target gene of interest. So we're always working within the endogenous levels of our gene, and we can TET regulate this on and off at any time. In the case of the TED-on system, this is the converse system where you produce an inactive transactivator protein, and only upon doxycycline addition will you actually inducibly knock down your target gene of interest. So you treat with DOX in these mice, you get GFP induction, hairpin, and then knockdown. So back when I was working in honours and then in the start of my PhD, we wanted to determine, well, what happens when you knock down Ikaros in the hematopoietic compartment of these mice? What is the general defects that occur when you do this? So what we did was we took an effective shRNA against Ikaros, and this was cloned into a TET-responsive construct here, expressing GFP and the Ikaros hairpin of interest. We developed transgenic mice that harboured this, and then crossed them on to a transactivator strain where you had hematopoietic specific, so from the VAV promoter, expression of TTA, and that means that in the absence of DOX, we had the knockdown of Ikaros throughout the hematopoietic compartment. And consistent with Ikaros mutant mice previously published, these mice actually developed a T-cell leukemia. So once again, TALL is generally the disease that emanates in these mice in the absence of any other influence. Now, what we saw was that these leukemias were often CD4 positive, CD8 double positive, so an immature stage of thymocyte development. So you had the aberrant expansion of these leukemic cells that were transplantable, and of course you could see extremely high levels of GFP, suggesting here that it was the Ikaros hairpin itself that was driving the onset of this disease. So one of the major advantages to using this system is the reversibility of the hairpins. So what we could do is we could take our primary T-cell leukemia and then transplant these into immunodeficient mice, and upon the emergence of disease in the recipient mice, we could either leave the mice untreated, so the transplant recipient would have sustained Ikaros knockdown in this setting, or we could DOX treat these mice and inducibly restore our Ikaros expression levels. So what I've done here is I've shown spleen-resident tumour cells from these mice, and you can see in the untreated mice, we have extremely high expression of GFP, suggesting that we're having Ikaros knockdown in these particular recipients. However, if you treat with DOX and harvest these mice at different time points, you can see the gradual loss of GFP expression. And then if we look by Western blotting at the Ikaros protein levels here in two independent primary T-cell leukemias, we can see the gradual increase in Ikaros protein. So here we have an effective system where we can restore the expression of Ikaros within established leukemia in vivo, which hadn't been done before. But we had to take a step back at this point because we were generating a T-cell leukemia, and this gene was often deleted in B-cell leukemia. So we had to think about that and determine whether or not we pursue this or we try to force a BALL. But what was really interesting to note was that in previous literature, it's been shown that Ikaros loss and Notch1 activation actually cooperate in the genesis of TALL. So Notch1 itself is a cell surface receptor on developing thymocytes, and it's critical for the commitment and development of normal thymocytes. However, in human T-cell leukemia, around 60% of human TALL cases actually harbour activating mutations in Notch1. And these occur in two key domains. There's the heterodimerization domain and PES domain here. And what this does is it actually results in ligand-independent signalling of Notch1, driving the expression of genes such as MYC, HES1, pre-TCR, and DELTEX1, which actually drive the proliferation and survival of developing thymocytes. And this is oncogenic. So these activated forms of Notch1 are so common in TALL that we thought we'll pursue this because perhaps we're modelling a more human-relevant disease than we thought initially due to this cooperation between Ikaros loss and Notch activation. So the first thing we did in this case was we took our Ikaros knockdown TALLs And then we conducted RNA sequencing and we looked at the Notch1 locus to determine whether or not we're also seeing activating mutations in Notch1. And what I've shown here is a schematic of the Notch protein, and we found that there were mutations in our knockdown TALLs. In this study, we actually concentrated on 3 of our primary TALLs, ALL 65, 101, and 211. And each of these had different Notch1 mutations, all predicted to be activating mutations and occurring at residues that were reminiscent of those seen in human T-cell leukemia. So this was extremely exciting to us because we saw we had a Notch-activated tumour, and now we could inducibly restore Ikaros within these to understand how Ikaros influenced this disease. Now, when we conducted Western blotting on these tumours, we saw that ALL65 itself expressed a full-length form of Notch1, whereas we saw in ALL101 there was actually a truncated form of Notch1, which was highly expressed in these cells. ALL211, however, had a truncated form of Notch1 again, but it was really poorly expressed, and this is relevant to later in the talk. So it's also been noted that in the previous Ikaros mouse models, when you have loss of Ikaros either through generation of a dominant negative or a null allele, the thymocytes of these mutant mice actually have elevated Notch1 expression, and also in their TALL cell lines, they saw this as well. But we had a very unique opportunity where we could actually look at how Ikaros was influencing Notch target expression in an in vivo setting in tumour cells. So we did exactly that. We took our RNA-seq data of the combined 3 T-cell leukemias, and on the y-axis we can see the log-fold change of all of the genes in the transcriptome of these mice. On the x-axis we see the average expression of these genes. And we find first and foremost when you can compare untreated mice where you have Ikaros knockdown versus mice that are dox treated in this case, we see that Ikaros is actually the most highly upregulated gene in our RNA-seq dataset, suggesting that the technology is working effectively and we can restore Ikaros robustly in all 3 tumours. Then if you look at the Notch1 target gene expression across all of the tumours, you see that in every single case you can get a reproducible suppression of Notch target gene expression. So for the first time we've been able to demonstrate in vivo that Ikaros can actually suppress the activity of Notch target genes. And then from some ChIP-seq data of normal double positive thymocytes using an Ikaros biotin mouse, Hiromi and Meinrad at the IMP were able to show that Ikaros actually bound these loci directly. So even though not the perfect cell type control, there's every chance that this suppression is actually direct, this is caused by direct Ikaros binding. So now we wanted to really ask the question, well, we have Ikaros knockdown tumours that are Notch activated, so how important is Ikaros suppression in the survival of these cells? So what we can do here is we can transplant disease and then wait for the emergence of the disease here at day 0. So you can see this CD4, CD8 positive population in the periphery of these mice. And then we can place them on dox for an extended period of time or leave them untreated, and then have a look at how the cells will respond to Ikaros restoration in the presence of these activating Notch1 mutations. What we find is that in ALL65 and ALL211, we get a significantly increased survival of these recipient mice when we treat with doxycycline, and this is coupled with the clearance of the peripheral tumour burden. So this is all from the peripheral blood of these mice. And we get a clearance of this peripheral burden. However, for ALL101, interestingly, we never saw that robust regression of the disease. Even if we restored Ikaros in this case, they still grew out. So it begged the question, well, why is this actually occurring? So first and foremost, we thought, well, perhaps ALL101 doesn't actually induce the expression of Ikaros when we treat with dox, even though we're using GFP as a proxy in this case, and Western blotting suggested it looked okay. We wanted to check by RNA-seq. What we found was that in all 3 tumours, they could all robustly induce the expression of Ikaros at the mRNA level. So then it led us to think, well, these are Notch-activated tumours, so perhaps there's varying amounts of Notch signalling in these cells. And that's what we saw. So in ALL65 and 211, we saw particularly low expression of Notch target genes, whereas in ALL101, there was particularly high Notch target gene expression. And even after an acute restoration of 3 days dox, you never could get these target genes back down to the levels of the regressing tumours. So it suggested to us the possibility that perhaps the level of Notch1 signalling influences the ability for a cell to actually regress upon Ikaros restoration. And you would have noted before that The DOX-treated mice actually do come down with the disease following treatment, and this is a GFP-negative disease, but a T-cell leukemia. So when you actually take these mice and you interrogate what's happening to them at the level of Ikaros, we see here that in the majority of these recipient mice that relapse, we have the emergence of a dominant negative isoform of Ikaros. And this suggests here that this is a means of actually circumventing Ikaros restoration through some type of genomic event, and therefore these cells will have a growth advantage. So that's just one mechanism by which these cells will relapse. So back to the question at hand, which was, well, is the repression of Notch1 responsible for that regression of TALL when we restored Ikaros? And one way to investigate this is to take the tumours that are responsive to Ikaros restoration and try to sensitise them with ectopic Notch1 expression. So you take your T-cell leukemia and you do retroviral infection ex vivo of either a non-silencing hairpin control ranilla, where we'd expect these infected cells, when injected into mice and then DOX-treated, to respond as we saw before with that initial regression and potentially a relapse. We can infect in a stable Ikaros hairpin as our positive control. So even when we attempt to restore Ikaros in this setting, we should still get stable knockdown of Ikaros in these cells, and they should have a growth advantage and not respond to that restoration. Then finally, the test infection would be overexpressing the signalling portion of Notch1, ICN1, then attempting to restore and seeing whether or not if a cell that's unable now to suppress Notch1, is that able to regress anymore? So what I've shown here is actually the peripheral burden of individual recipient mice following the induction of doxycycline at disease detection. So in the black lines are the negative controls, and we can see in 65 and 211, we get an initial regression of the disease in the periphery, and then, as I showed before, they will eventually bounce back, often with a dominant negative. However, when you infect in ALA 101, once again grows out, so that's just as we'd seen before. With the stable Ikaros knockdown, we get the continued growth of the tumour cells independent of DOX treatment. As you would expect here, we're not able to restore Ikaros anymore. And this is also seen when we ectopically overexpress Notch1 in these normally responsive tumour cells. So collectively, what this suggested to us, and it's supported here by survival analysis, is that Ikaros restoration suppresses Notch target gene expression, leading to the regression of the T-cell leukemia in these transplant recipients in vivo. But we wanted to now take this into a human setting. We'd been able to demonstrate that relationship in mice. So we wanted to see, well, what's happening in human T-cell leukemia with activating mutations in Notch1? And with the help of Eliza and Tom Tramaci at NYU, they were able to provide us with a lot of microarray data and a few really nice in vitro experiments to explore this idea. So Tom actually took 10 independent human Notch-mutated T-cell leukemia cell lines and just looked at the expression of Notch target genes versus the expression of Ikaros itself. And what he was able to show here was that Ikaros expression inversely correlated with Notch1 target gene expression, consistent with what we'd seen between that antagonism between Ikaros and Notch1 targets. Furthermore, Eliza was able to show that in T-cell leukemia, human TALL cell lines in this case, CUT-L1 and HBB-ALL, these are both Notch-activated T-cell leukemias. When you inhibit Notch target gene expression using pharmacological inhibition, so that's gamma secretase inhibitor here, so you inhibit Notch1 in this case, and we actually saw de-repression of Ikaros mRNA. Furthermore, if you continue to enforce the expression of activated Notch1 in these cells, you actually get repression of Ikaros mRNA in these cells. So overall, what this suggested to us is that activated forms of Notch1 itself can actively suppress the mRNA expression of Ikaros, and we'd been able to show in our mouse model that Ikaros itself could suppress Notch1 target gene expression. So it presents this paradigm, at least at the mRNA level, where we see this mutually antagonistic relationship between Ikaros and Notch1. And what we believe is a possibility here is that we don't see really any, well, there's around 5% or so mutations in Ikaros in human T-cell leukemia. However, 60% of cases have activated forms of Notch1. So perhaps one means by which activated Notch1 can provide an environment permissive to TALL transformation is through the suppression of genes such as Ikaros, which would otherwise repress the expression of Notch1 targets. However, this still has to be proven at the protein level. We're yet to demonstrate this and potentially xenografting. So just to summarise the first part of this talk, It's quite clear from our studies that Ikaros knockdown drives a notch-activated T-cell leukemia, and when we restore the endogenous levels of Ikaros in these T ALLs, you get suppression of Notch1 targets, which may cause a transient leukemic regression. We believe using RNA-seq and ChIP-seq, we've identified some really nice direct Ikaros targets in vivo, and that activated Notch1 itself can override that Ikaros restoration-mediated regression. Finally, in human T-cell leukemia cell lines, in this case, we've shown that activated Notch1 can suppress Ikaros mRNA. But that's all in T-cell leukemia. So as I mentioned, Ikaros deletions are quite a rare event in this disease. So we wanted to actually model a more clinically relevant type of disease, which is human B-ALL, where Ikaros has really been well documented. And as I mentioned before, the disease subtypes of the PH-like and BCR-ABL B-ALLs have a particularly poor prognosis under standard chemotherapy. And this is compared to all other B-ALLs. And really interestingly, it's been shown that the deletions in Ikaros are so enriched in these high-risk subtypes. So BCR-ABL positive, around 80% of them have an Ikaros deletion. PH-like, it's more like 70%, whereas all other B-cell leukemias, it's 15%. And this is really interesting as well with the fact that Ikaros mutations will independently predict poor treatment outcome. So they're often enriched at relapse B-ALL, they predict resistance to standard chemotherapy, and more recently it's been shown that Ikaros mutated patients in pH-positive disease respond poorly to targeted imatinib therapy. This is shown here, this is just standard chemotherapy in pH-like disease, where we see Ikaros alterations will predict a poor outcome independent of pH-like. Now, what's interesting to note about these high-risk subsets, so BCR-ABL positive disease and pH-like disease, is that although quite rare in children, so only 15% of childhood B-cell leukemia are pH positive or pH-like, with increasing age, actually the proportion of these particular subtypes increases dramatically. And we know from many studies that older patients respond far less favourably to chemotherapy, suggesting this may be a major driver of that poor response in this group of patients. And this just reiterates the fact that these particular subtypes have such a poor response. So the questions are, firstly, well, how does Ikaros cooperate with these particular lesions to drive B-cell leukemia? We know just how enriched Ikaros lesions are in these groups, so why is this happening? And then why are these patients so resistant to chemotherapy? So we conducted a very similar approach to the T-cell leukemia story that I showed earlier, where we had a hematopoietic knockdown of Ikaros using VAV-TTA, but now we cross these mice to a transgenic mouse which stably expressed BCR-ABL from the metallothionein promoter. And in 1990, Nora Heisterkamp was able to show that these mice actually developed a B-cell leukemia at around 90 to 100 days latency. So it was an ideal model for looking at this interaction between Ikaros and BCR-ABL. Furthermore, it's been shown more recently that Ikaros hypomorphic mutants can accelerate the onset of this particular mutant. So we took our mice and we generated two progeny. We had BCR-ABL progeny with the VAV-TTA, so they're our control littermates, and then we had a triple transgenic mouse which harboured BCR-ABL, transactivator, and also the TET-regulated Ikaros hairpin. So the control mice generally came down with the disease around 80 days, at around 80 days, and this was a pre-B-cell leukemia. And furthermore, Ikaros knockdown accelerated the onset of this disease, so they would come down with the disease around 36 days. So this is really interesting to us because it showed that, firstly, hairpins can effectively disable the tumour suppressive function of Ikaros in the B lineage, and we're recapitulating that interaction between BCR-ABL and Ikaros loss in this particular mouse model. Immunophenotypically, these two subtypes were, two groups were indiscernible. So for the duration of this talk now, I'll really use CD19 positivity and IgM negativity as the marker of immature B cells when tracking the burden of these mice. But at the RNA-seq level and immunophenotypically, they appear to be large pre-B cells. Control B-ALLs obviously don't express any hairpins, so they're GFP negative, whereas the triple transgenics have really high expression of GFP. So we know that the hairpin itself is facilitating this disease. So once again, using this technology, we're able to transplant this disease, restore Ikaros, and just see, well, what's the influence of Ikaros specifically in the survival of these leukemia cells. So the tumours themselves are Li5-2 positive, these primary B-cell leukemias, whether control or knockdown. You can transplant these into Li5-1 positive RAG-deficient mice, and then once again either leave them untreated or treat them with dox. So in the Ikaros knockdown tumours, you can restore Ikaros in vivo in the established disease. So what we can see here is, once again, splenocytes from these recipient mice. We see in the untreated, we get really high GFP expression. If we harvest these mice at different time points, post-dox, we get gradual reduction in GFP. And then we can see here that in the control leukemias, when you harvest for Western blotting, there's no responsiveness to dox when you look at Ikaros levels in the tumour cells. Whereas in the knockdown tumours, we can see a robust induction of Ikaros. And it's interesting to note here that the level of Ikaros knockdown is actually quite poor. It's around 50%. Yet we still see such a dramatic acceleration of the disease, showing just how sensitive B-cell progenitors are to any perturbation of Ikaros during leukemogenesis. So now we wanted to leave them on the shelf and see, well, what happens if we restore the endogenous levels of Ikaros in these tumour cells? How do these cells respond to that? And it was quite striking. So this particular Kaplan-Meier curve shows 6 different primary B-cell leukemias with 2 to 3 recipient mice per group. And what we found was that if you treat them with dox, and these are days post-detection of leukemia in these particular mice, we generally wait for the proportion of CD19 positive IgM negative cells to exceed 5% of the peripheral lymphocyte burden. We find that in untreated mice, they come down with the disease at around 19 days post-initiation of the experiment. However, when you treat these mice with doxycycline, we get a profoundly increased survival of the recipient mice treated with dox, and they actually survive out to 113 days median survival. And a few of these mice are still on the shelf today, extremely happy with no peripheral burden. So what this suggested to us is that by restoring the expression of Ikaros in established disease, these cells are really sensitive to the level of Ikaros for their survival. And it must be noted that these tumour cells themselves, even though we've perturbed Ikaros in this case, they all have stable BCR-ABL expression. So it suggests here that they're critically dependent on Ikaros loss for their survival. Now, if we look at the peripheral burden of these mice in the untreated cohort, at day 0, we have quite a high burden in this particular recipient, and if you leave it off dox for 19 days, it'll eventually grow out. However, if you treat a mouse with a similar peripheral burden with doxycycline at this time point, you can see the CD19 positive IgM negative population here essentially dissolves, and it suggests that there's clearance of the tumour burden in these mice. Next, we want to look at secondary lymphoid tissue, not just the peripheral blood. We find that in control mice, either untreated or dox-treated, we get an effaced splenic architecture, suggesting you have a complete infiltrate of leukemic cells in these mice. This is very reminiscent of the untreated Ikaros knockdown tumours here. We can see, once again, very similar to these controls. However, when you treat with doxycycline and harvest the spleen after, say, 14 days, when we see clearance of the leukemic burden, we can see that there is now no longer that CD19 positive IgM negative population in these spleens, and you actually have some type of normalization of the splenic architecture in these immunodeficient mice. So that transgenic cross, it actually took quite a bit of time, and Ross would tell you that I'm really impatient at the best of times, so I wanted to generate alternate models to look at this interaction between Ikaros loss and BCR-ABL. So I won't actually dwell on this too much. Needless to say, we took a retroviral approach in this case, where we have TET-regulated hairpin mice. We harvest the whole bone marrow of either the knockdown or vanilla mice, and infect in stable expression of the TET-off transactivator and also stable expression of BCR-ABL itself. And when you reconstitute these mice, we can see that the control leukemias in black actually come down extremely fast. It's probably due to multiple integrins of BCR-ABL in these particular reconstitutions. And when you knock down Ikaros, in addition to that, you have an accelerated disease. This is readily reversible, and also when you're restoring these transplant recipients, you do get clearance of the peripheral burden. So what I've shown there is that there's an alternate model demonstrating that interaction between Ikaros and BCR-ABL. So now we wanted to define, well, what are the critical target genes mediated by Ikaros that drive not only the acceleration of B-cell leukemia in this case, but also the restoration phenotype that we see when we restore Ikaros, the tumour regresses. So what are the genes that are mediating these two particular phenotypes? So the first analysis we did, and this is with the help of Gordon Smythe and Yifeng Hu, who basically helped us with every bit of bioinformatics in this and were extremely patient with us throughout these analyses. We compare, firstly, in the acceleration group, you take control B-cell leukemia and you compare them to the Ikaros knockdown leukemia. So you're looking at the transcriptional difference between the accelerated group here and also the control group. So what are the genes mediating this? Next, we took untreated leukemias, so this group here, and we compared them after a transient Ikaros restoration to the Ikaros restored group. And we're trying to identify, well, what is Ikaros setting up transcriptionally that will mediate the regression that we see in this dox-treated group? And this is what the data looks like ranked on moderated T statistic here. We see on the y-axis we have genes differentially expressed between untreated and dox-treated leukemias where we see restoration in the Ikaros knockdown group. And then on the x-axis here we see the differentially expressed genes between the control B-cell leukemias and the Ikaros knockdown BALLs. And first thing you'll notice is there's a nice correlation between these groups. So there is co-regulation of some of the target genes here mediated by Ikaros. We then identify two major groups using a false discovery rate of less than 5%. We have an Ikaros activated group up here and an Ikaros repressed group. So there are 103 genes here that have particularly low expression in the Ikaros knockdown B-cell leukemias versus control leukemias. And then when you actually restore Ikaros in these cells, their expression is also reactivated. A repressed gene, however, has particularly high expression in Ikaros knockdown tumours when compared to control leukemias, and restoration of Ikaros will drive their suppression. So that's also shown here for clarity's sake. We see here in the Ikaros repressed group, you have particularly high expression in the knockdown group compared to control. You restore Ikaros and the expression of these genes starts to drop. In the activated genes, we get lower expression in the knockdown group compared to control. You restore expression of Ikaros and their expression seems to go up. Interestingly, once again, Ikaros is the number 2 activated gene, so we're happy that the system's working correctly in this case. So now we believe we've generated some really high confidence Ikaros target genes in murine B-cell leukemia, but we wanted to take now a non-biased approach with more recently generated human B-cell leukemia data to identify genes that are conserved between human and mouse and Ikaros regulated, because it's nice to generate mouse targets, but really we're here to figure out, well, what's happening in human BALL in these Ikaros deleted patients. So Catherine Roberts and Charles Mulligan have been fantastic with providing us with data, and in Catherine's recent paper in New England Journal of Medicine, they were able to conduct RNA sequencing of 289 human B-cell leukemia patients, the largest RNA-seq study on BALL to date, and 252 of these samples had known Ikaros status. So we took Ikaros mutated B-cell leukemia patients and compared them transcriptionally to Ikaros wild-type patients using a false discovery rate of less than 1%, and we looked for genes that were expressed in over half of the B-cell leukemia patients. We then compared this to our shRNA model where we had our Ikaros activated and repressed genes I showed before, and we overlapped the two differentially expressed groups, so Ikaros activated and repressed in human, Ikaros activated and repressed in mouse. And what we found was not phenomenal at the time, but it was still really interesting to us because we found that there were a very small number of genes that were conserved between human and mouse here. We had Ikaros repressed genes, so we had 7 of these, and we had 3 Ikaros activated. And what this suggested to us is potentially the species difference between human and mouse is really exacerbated in this study, or because we actually believe we've done quite a nice job of identifying Ikaros-regulated genes in mouse. We're effectively recapitulating the interaction between BCR-ABL and Ikaros loss in this model, and we're using all of the most stringent cutoffs we can to generate our list. However, this is what came out of it. But what was interesting to us is that many of these genes had previously been implicated in leukemia before. And I'll just dwell on one of these, CTN and D1, for the end of this talk, to try to convince you that we pulled out some really nice conserved genes between human and mouse. So CTN and D1 encodes P120 catenin. And in our murine dataset, it's actually the second most highly ranked repressed gene. You can see in the control B-cell leukemias here, expression is quite low. And in the knockdown leukemias untreated, it's particularly high until you restore Ikaros and you can get its levels back down to normal. In human ALL, we see in the Ikaros repressed group, this gene actually ranks number 17 out of the 645 repressed genes. And in mutant cases of Ikaros-deleted BALL, there's around a twofold increase in the expression of delta-catenin in these groups. At the protein level in our murine BALL model, we're able to show that in the knockdown leukemias untreated, they have elevated levels of P120 catenin compared to control. When you restore Ikaros, we get the downregulation of expression of P120. And then in independent primary, if we sustain Ikaros restoration here, you can see the levels of Ikaros going up. We get a continued degradation of P120 catenin expression in this case. Now, P120 catenin is really interesting to us because it is involved in cell-to-cell adhesion, and also it's been shown to activate the beta-catenin signaling pathway. And more recently, it's been shown that activated beta-catenin is a feature of many relapsed childhood B-cell leukemias. Also notably, an older study looking at the expression differences between BCR-ABL positive BALL compared to all other ALLs showed that CTNND1 was amongst the most highly upregulated genes in this group, yet they weren't able to make the link to Ikaros deletions in this case. However, we know that Ikaros deletions are enriched in this subset. So what that suggested to us is this is a bona fide Ikaros target gene in both human and mouse. Now, another group that's really interesting to look at is this relapse group. So if you leave the mice on doxycycline for a long time following transplant, you get a regression initially, and then a proportion of these mice will actually come down with the disease, a B-cell leukemia eventually. This is often CD19 positive, IgM negative, and there's no expression of GFP. So the hairpin's being effectively shut off, yet the disease is finding a way to re-emerge and circumvent Ikaros restoration in this case. So we looked at the transcriptional profile of our relapse disease to identify, well, what's happening to these cells? Why are they bouncing back with BALL? And what we found is when you compare in this primary B27, for example, the relapse disease versus the untreated leukemia in recipients and docs treated, there was more of a relationship between the relapse disease and the untreated disease than those treated with Ikaros restoration or doxycycline. And specifically, if we look at CTNND1 in this case, we get the gradual loss of CTNND1 expression over dox treatment, but then at relapse, you get the restoration, if you will, of CTNND1 expression, suggesting that this gene may be part of the transcriptional program driving relapse in these cells. Furthermore, we had 3 different relapses from 3 different primary mice. We RNA-seqed them all, and what we found was that this was a consistently upregulated gene at relapse. Now, we have this data set of human RNA-seq. We want to see what CTNND1 behaved like in this group. So we took RNA-seq from our 289 patients, and we plotted the expression of CTNND1 across all patients, and then assigned a rather arbitrary cutoff of log2 FKPM greater than 4 in this case, and called these patients CTNND1 high, and these patients CTNND1 other. And what we find is just, consistent with the way that we identified CTNND1 as an Ikaros target, the other patients had around 40% Ikaros mutations, whereas the CTNND1 patients had a high proportion of Ikaros mutations, as you would expect with the way we generate these target genes. Furthermore, in the CTNND1 high patients, they have an inferior event-free survival of 52%, 5-year event-free survival, compared to CTNND1 other patients, reminiscent of Ikaros-deleted B-cell leukemia. So once again, we think we've demonstrated here that we found a really nice Ikaros target gene that may be potentially druggable as it's highly expressed in Ikaros mutant BALL versus wild type. So now we're currently doing experiments to actually address this, and one way we can do this is actually by inducibly knocking down the expression of CTNND1 in our Ikaros knockdown BALLs and determining, well, whether or not high CTNND1 expression is associated with survival of the tumour cells. So if you knock it down, as in like a pseudo-drug treatment, if you will, do these cells respond to this? So Grace and Ollie, so Ollie is a Europe student who joined the lab in November, and I've been taking care of Ollie. He's been really great for the lab and works extremely hard, and everyone who knows Grace knows just how hard she works. Grace and Ollie, actually, when I was away gallivanting around the world, they were actually able to optimize a couple of hairpins against CTNND1 for me. And using an AML cell line, they're able to show that these 2 particular hairpins had really nice knockdown of CTN and D1 compared to non-silencing, and we use a positive control of MYC in these experiments. And at the Western blot, you can also see these are very nice hairpins. Now, the way we can actually address whether or not this gene is critical in the survival of Ikaros low BCR-ABL positive BALL is to generate what we call a TET-on competent BALL. And to walk you through this, we take black 6 bone marrow and we co-infect stable expression of our TET-on transactivator protein linked to shRNA against Ikaros. And this is co-infected with BCR-ABL in this case, and we reconstitute mice. And what we should see here is the cooperation between BCR-ABL and Ikaros knockdown in the emerging tumour cell population, and therefore in all of the tumour cells of these mice, they should be expressing RTTA. Then you can take these tumour cells ex vivo and infect in a TET-responsive hairpin vector, which will allow for the inducible knockdown of any target gene of interest in the established tumour cell. So this vector, just to run you through it, is called the TRMPV vector. And we can see here when you actually activate RTTA with doxycycline, you get binding of the TET-responsive element here, driving the expression of DS-RED and a hairpin against CTN and D1 in this case. And then all infected cells in this tumour population will express venous as a marker of infection in this particular group. So we can take the infected tumour cells here, inject them into mice, and then induce doxycycline to turn on their CTN and D1 hairpin and see, well, is this gene critical for the survival of these cells? And this is, so what I've done is I actually did the infection ex vivo. I took a small cut of cells, plated them on OP9, and then the remaining cells I injected into the recipient mice. So from the OP9 co-cultures, we can see in the untreated primary B-cell leukemias infected with this TRMPV vector, we have a high proportion of the cells infected with the vector, so they have stable expression of GFP. However, when you induce doxycycline for 3 days in these cells, you can see the robust induction of DS-RED, which would be the hairpin against either Ranilla, MYC, or CTN and D1 in this case, demonstrating that we've generated a nice TET-on competent B-cell leukemia, which we can interrogate some of these repressed genes. This is what it looks like by qPCR, and this is just early days, so it's only a couple of experiments in the average of those. However, we're really excited about this because elevated CTN and D1 produce, it really sets up a nice druggable target in these leukemias, and if it is critical for the survival of these leukemias, it tells us a lot about how Ikaros mutated BALL behaves compared to Ikaros wild-type B-cell leukemia. So for the future directions of my PhD, for the remainder of my PhD and my time before I head over to New York, I'll be firstly trying to wrap up these experiments and determine, well, are these repressed genes critical for the maintenance of B-cell leukemia? And also I'll be doing a couple of positive selection experiments whereby you take your Ikaros repressed gene or your activated gene in this case, and you can either overexpress this gene in combination with BCR-ABL and ask the question, do these lesions cooperate with one another in the pathogenesis of this disease? Or you can knock down your gene of interest and see whether or not this happens as well. Another thing we can do is similar to our T-cell leukemia study, we can capitalize on the fact that our leukemia cells will regress upon Ikaros restoration. So we can stably knock down our activated gene or overexpress our repressed gene, infect those into the tumour cells, inject them into mice, and then challenge them with Ikaros restoration and see whether or not these cells will selectively grow out, inferring that they have a critical role in that regression phenotype. So that's what I'll do for the rest of my time here. Just to summarise, we've been able to show that knockdown can effectively disable the tumour suppressive function of Ikaros in the B lineage in combination with BCR-ABL, and that by restoring Ikaros, these cells are extremely sensitive to any perturbation in Ikaros levels. So we know that Ikaros loss is critical for the survival of these cells, and when you restore, despite constitutive BCR-ABL, the cells will still die. We've been able to show that by combining human and mouse, we can pull out a few conserved genes, if you will, and that some of these genes actually do play out as genes related or previously implicated in B-cell leukemia, for example, CTN and D1. And this predicts an enrichment for Ikaros mutations. It also recapitulates the event-free survival we see with Ikaros mutation. So now the rest of my time will focus on actually testing these genes for functional relevance in vivo and whether or not they are druggable. So it just leaves me to thank some people before I go today. The Dickens Lab, we're a very small crew, so for the most part of my PhD, there was only 4 of us. So there was Ross, my supervisor, and I thank him so much for even giving me a job in the first place. He didn't really know what I was about. I just rocked up and then eventually gave me a job. And it's been fantastic. But he's a phenomenal guy, and we've built a really nice relationship over the years. He's great to talk to, and we've been through a lot of really good times, but some pretty bad ones as well. So it's nice to have someone that you can really rely on to talk to about these issues back and forth, and it's been a phenomenal experience for me. Mark McKenzie is an exceptional scientist. He's been through a lot of adversity with his project and bounced back phenomenally. So a really great guy, and I've learned so much from Mark over my time here, particularly how to deal with the bad part of science and results not going well. And he's finally getting some amazing results, so it'll be great. Grace is leaving us soon, which is heartbreaking, because Grace came into my PhD about a year or so in, and there was a lot of testosterone flying around in the lab. And anyone who knows Grace, she's calm blue ocean. So it really settled things down. The next generation of Dickens Lab members, Ollie, Minhee, and Amelia, are going to be fantastic, and they're really setting up a nice program at the ACBD with Ross. Gordon and Fang have been extremely patient with us over our time, over my time at WEHI, and I can't thank them enough for their help. Everyone in molecular medicine, it's a great place to work. It's really fun. I'm very annoying, so they've been really great in putting up with me, and particularly Tracy Wilson, who isn't here at the moment. She's not too well, but she was really instrumental in setting our lab up. This is taking a long time, so just settle in. Ben Kyle actually has been fantastic in giving me a home whilst the lab move, and also outside of work, he's an extremely good mentor to me. Giannis's lab at NYU, I'll be moving into Giannis's lab in November, and Louisa was actually the first supervisor I had at WEHI, and I'll be reunited with her, so we'll get the band back together over there. It'd be nice. And then Tom Tremachi's been fantastic in the TALL stuff. Catherine and Charles for their human data, HRF for all their help, Hiromi and Meinrad for their support, and Ikaros Bites and mouse data, Steve Wilcox at systems biology. Ketty and Carolina have been exceptional animal techs and friends of mine, so I can't thank them enough for all their help. Lockie, flow cytometry. I've got a fantastic PhD committee, so Andrew Roberts, Marnie Blewett, Andreas and Tim, particularly Tim, thanks for coming from Monash today. But they've been great in their guidance. Leukemia Foundation have supported me in my on and off field endeavours at WEHI, so I can't thank them enough for that. Edith Moffat and NHMRC for their financial support. WEHI for giving me the opportunity to do a job I love to do. It's been great for me. My family and friends, so thanks for coming and listening to 15 minutes of jargon. It's been great to have you here. It's a lot of support. And as I said, Grace is on the way out. It's shattering. I'm really disappointed about it. So I want to thank you and wish her all the best. And thanks for your time today, guys. Cheers.
Any questions for Matt?
Probably just a bit of clarification. So when you characterize your TAL, you characterize them as CD4 and CD8 positives. Are you suggesting that this TAL formed before it's entering thymus or after it's exited thymus?
So we believe it's forming after it's input into the thymus. So it is a thymic disease, for one. It's also been shown in Ikaros mutant mice that if you thymectomise the mice, they don't get T-cell leukemia. So we're fairly confident that this is a disease of thymic origin. Whether CD4 and CD8 actually recapitulate the stage of development that's transformed is another question, because, for example, in the Alamo-2 mouse, for example, Matt McCormick's had to see a block at the DN2 stage, yet what emerges is often later in development than that. We see the same thing. Often there's a DN4 accumulation in the pre-leukemia, however, a DP leukemia emanates. So whether or not it's a true reflection of the stage.
No, have you tried to characterize that TLL, like the T cell, the CD4, CD8 positive cell with B cell marker or something? Because I know that Notch1 knockout hematopoietic system will develop B cell instead of T cell in the thymus.
Sure. This is actually a Notch active situation though. So a Notch active mutant, conversely, will cause ectopic T cell production in the bone marrow of mice. So we believe this is certainly a T cell leukemia. However, yeah, we haven't done, I think it's pretty clear from this that it's TAL.
So apologies, Matt, for I got this asked backwards, but your 101 tumour, when you re-expressed Ikaros, it didn't respond, and you think that's because it had this mutation in Notch and high levels of Notch?
Sure.
Is that right? Was that the same tumour that when you did Western for Notch, there was no expression?
Oh no, sorry, that's actually the one just next to that where it has a really high expression of the truncated form. Yeah.
Does Ikaros repress other Ikaros family members?
Yeah, it's a good question. So there's a bit of debate in the field whether or not Ikaros actually regulates islet function. So often people implicate other family members of Ikaros, Islet, Helios, and whatnot, as functionally redundant members of the Ikaros family. Now, Meinrad Boslinger's recent work using a CD79 beta-Cre deletion of Ikaros in the B lineage has shown that perhaps that functional redundancy is not so pronounced. However, conversely, the Ikaros dominant negative mouse has a profoundly different phenotype than the Ikaros null mouse, where the null will not affect other family members per se, so it would allow them to exert a functionally redundant role, whereas the dominant negative mouse in theory should sequester their function. So they're profoundly different. I don't see from RNA-seq data a consistent elevation or repression of
Just in the figure you showed, you had the IKZF4, you had upregulation of IKZF4.
Yeah.
I was just wondering if they had overlapping target genes or consensus binding sites in DNA.
Sure, yeah, I know that Ikaros and Islet share consensus binding sites. However, that's EOS. I'm not really sure what it does. Is it EOS or Pegasus? I can't remember. It's one of them.
Just Greek, it's good.
It's one of the great ones.
And the Ikaros targeted genes that we had very low overlap between human and mouse. Do you think, did you try and loosen the restraints to see if you were just sort of selectively losing different genes at different cutoffs? Was that throughout the whole population?
Yeah, that's a really good question. So we did a few things. So we actually looked within subtypes of ALL. So that group is actually across all BALL subtypes. And we tried to determine what the influence was of Ikaros within, say, for example, pH-positive disease. We lost a lot of statistical power there because we only had 30 patients, so that was a problem. And in an outbred population, it's a mess at that point. Yeah, it's a really good point. Perhaps we could be far less stringent, but we thought we'd try to go for the really high confidence ones initially and see how they read out. However, yeah, as you may have noticed, we used an FTR of less than 1% in the human, but 5% in the mouse. I mean, perhaps we loosen the purse strings on the human and see what happens.
I'm a bit surprised that you didn't mention any mechanism for killing these cells after you restore Ikaros.
Sure, it's a good question. So in the T cell leukemias, I do see apoptosis, but in the BALL, I haven't been able to see it. They get cleared so quickly that I just can't really pick them in the peripheral blood. But I'm sure in vitro, they actually behave. This is an interesting point and maybe a bit of an aside. But ex vivo, when you restore Ikaros in the presence of exogenous IL-7 on OP9, they actually don't regress in ex vivo. However, in vivo, when you put them into mice and you restore Ikaros, they regress, and presumably they may undergo some form of apoptosis. I'm not sure. However, why this occurs, I really don't know. Perhaps that would implicate cytokine signaling is important in their survival in the ex vivo system. One interesting thing that comes out of this is in the spleen plots you saw, there was the emergence of an IgM positive population that sat in the spleen after restoration. So perhaps similar to what Grace has seen in the PAX5 situation, where you see actually a differentiation of cells through to a more mature cell type. Perhaps this is a mechanism by which you get tumour clearance. Maybe there is apoptosis coupled with some form of differentiation. This is purely speculation. Charlotte?
As a sort of follow-on from Melissa's question, the bioinformatic analysis of the patient cohort, is it possible that, if I'm not mistaken, the way you did it was to say which genes were expressed with an FDR or whatever in at least 50% of patients.
Just expressed in at least 50% of patients.
Expressed. Could you do the reverse and say, well, what does my mouse tumour, which is there perhaps a sub-selection of the patients my mouse tumour looks more like compared to the others?
All right. So yeah, we that's actually a good point. Yeah, we haven't done that yet. So that's a really good idea. So perhaps taking a non-biased approach and comparing our profile of our activated repressed in mouse just to the human setting. It's a really good point. We haven't done that yet.
Just on, sorry.
Yeah, go ahead.
Just on those kind of questions again. Is that difference between human and mouse, is that like common? Like, should people that work on mouse know that that's not really like what the human is? Or is this specific to what you're doing?
Sure. No, it's a really good point. Right? I think, in defense of what we've done, I think we're one of the first ALL studies to actually compare in a non-biased fashion regulated genes in human and mouse. Also, there's not many data sets that strong out there for a lot of diseases. I mean, that's a lot of RNA-seq data for a lot of patients. So, I don't know whether or not, I think the genetics of the mouse can really model what's happening in human disease. So we see Ikaros loss cooperating with BCR-ABL. That's what happens in humans. However, whether or not you can get down to the nitty-gritty gene targets in an outbred population of humans versus inbred mice, that may be the question. But I think it does not discredit the mouse as a model of leukemia. What it does suggest is perhaps we have to be a bit more careful when saying, well, this gene is going to be critical for treating human leukemia when you pull it out of the mouse. But look, we're just getting into this now, so who knows? I mean, what's the precedence? I don't know. Andreas?
Before the BCR-ABL driven leukemia, why do you have to add IL-7 or other cytokines in vitro? Because BCR-ABL was discovered as an oncogene that makes cells growth factor independent.
Sure, yeah. I think it was initially, so that's a really great question because it will hyperactivate STAT5, which a lot of the cytokines will converge upon anyway. I'm not really sure. I just find that if I plate my Ikaros knockdown tumours on OP9 alone or off stroma, they will die. I don't really understand why that is, but perhaps I should pursue the ex vivo experiments a bit further because they're pretty interesting. That's a good point.
All right, very good. We'll leave it at that. It's 4:05, so it remains to thank Matt very much for his time.